You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you. Hello, this is Richard Jacobs with the Future Tech Health Podcast, and I have Dr. Richard McCulloch, a newly minted professor at the University of Glasgow. So, Rich, thanks for coming. How you doing?
I'm very well. Thank you very much for inviting me on your podcast. It's a pleasure to speak to you.
Yeah. So tell me about your research. What are you working on?
So the research that goes on in my lab is interested in the biology of the genome of cells and organisms. So what we're, the repository of all the information that's needed to generate proteins and RNA and so forth that drives cell function. In particular, what we're interested in is trying to understand how that genetic material is protected from harm and how it's transmitted in the process of cell division. So how it's copied and moved from cell to cell. This is a pretty wide field that we work in, and I guess most people would choose to study this in human cells because that's what makes us up. But what makes us maybe a little bit different, the niche that we're in, is we choose to study these processes not in human cells but in parasites that infect humans and cause disease in humans. And we've chosen to focus really in two different parasites. One of them is called the trypanosome. It's mainly found in Africa, and it causes a disease in humans called sleeping sickness. And the other parasite is another single-celled eukaryote, which is called leishmania, and that causes a very different disease. Well, in fact, it causes a number of different diseases, and it's got a much more widespread worldwide distribution than trypanosomes.
What are the vectors and mechanisms of action of a parasite? Does it actually integrate with our DNA, or does it keep its own, and does it take over cellular machinery like a virus? And what are the mechanisms of action of a parasite?
Well, so that's a good question. So one reason that we're interested in these two different parasites is that they've got quite different mechanisms of action. So what the common feature of these two parasites is that they infect humans both through the bite of a blood-feeding insect. So in the case of trypanosomes, it's the tsetse fly. In the case of leishmania, it's the sandfly. Both of them then go into the bloodstream, but then they do something really very different from each other. So trypanosomes, they live just in the bloodstream, so they don't infect any of our cells at all. They just live in the bloodstream, and they have to survive in there. And they've got a real problem with that because when they're in the bloodstream, they're confronted by our immune response to them all the time. So they have to come up with a strategy to try and cope with that constant immune barrage from us. Leishmania do something very different. What they do is they infect our cells. So they go into our cells and they live inside the cells. And that means that they have to be transmitted from cell to cell. And one reason possibly they do that is because that provides them a means to somewhat hide from the immune system rather than being constantly exposed to them. So the fact that these parasites do things similar in the sense that they're both transmitted by flies, but once they're in us, they do very different things, allows us to do comparisons about how the parasites operate and how they compare with each other.
Is the goal of a parasite to take a permanent residence inside the host, or is it to just be there long enough that it can lay eggs or whatever it is and spawn new life and have that life feed off the organism and then leave?
Again, it's a good question. So these are both of these parasites are kind of interesting for us because they're just single cells, so they don't make eggs or anything like that. They don't have complicated developmental biology like I don't know, something like worms or something like that you might imagine. But the goal is just as you say. So the really, as far as we can understand, the goal once they're in a host is to live and prosper on that host for as long as it takes them to be transmitted onto another host. And in both these cases, what that almost certainly means is be able to survive in the host long enough that when another fly comes along and feeds on that host, it then takes up the parasite and it kind of gets passed on to another host. So they're parasites because they're completely reliant on us or other animals for their survival. So their growth needs us, nutrients from us, for instance, and that as a result of that they do cause severe disease and they can potentially cause death, but it's a byproduct of their transmission biology.
Well, is the goal, like with the C2 fly, the fly is not deliberately infecting us with the parasite. The parasite just happens to use the fly as a host, and then because the fly will land on us, it'll go into us, or is it?
Yep, that's exactly right. So we often talk about, to give you another example of this kind of parasitology, I guess more people have heard of malaria that's transmitted by mosquitoes, and people will often say that mosquitoes are the biggest, one of the biggest causes of disease. But in reality, it's not the mosquitoes that's doing that. It's the fact that some of them happen to carry Plasmodium, which causes malaria disease. And it's exactly the same in the case of trypanosomes or leishmania. So the tsetse flies or the sandflies in the case of leishmania, what they are is just a vehicle that's been exploited by the parasite so that it can move from mammal to mammal. And a question that often comes up in the field is just what is the evolutionary history way back in the past? Did they start out as parasites of flies, or did they start out of parasites as mammals, for instance? And how did the two of these things get linked up? It looks as though the evidence is that they probably started in flies, but it's not absolutely certain.
So I was just thinking about malaria. Has anyone tried to make mosquitoes resistant to malaria so they can't be carriers of it? It seems like all the focus is making people, resistant to malaria or, killing all the mosquitoes. But what about making the common carriers of this stuff not compatible with these?
Yes, that's exactly those kind of experiments is probably too small a word for those kind of projects are being attempted now. So people are attempting to genetically modify mosquitoes, for instance, to try and come up with strategies that means that if, for instance, they might not be able to take up the malaria parasite, or when they take it up, it might not be able to go through the complex developmental program it finds within the fly. And as you say, that would, if that could work, that could potentially be a very productive way of trying to prevent the transmission of any of these parasites at all. The same kind of work is not anywhere near as advanced in trying to understand trypanosomes or tsetse fly function, sandflies, and that's partly because of the disease burden. Malaria is a much bigger disease. It's also because we probably have a better understanding of the biology, genetics and ecology of mosquitoes than we do of tsetse flies or sandflies. But probably in years to come, that will come. Yeah.
So what are the mechanisms by which these two parasites go into us, and why don't we create an immune response to get rid of them?
Okay, so that's really the kind of the real reason actually that I got interested in this area. You're absolutely right. You would imagine that we've got an incredibly sophisticated system for immunity. We're very, very good at fighting off infections. So an important question then is, how is it that any parasite is able to get into a host, is able to grow, and in the case of the trypanosomes, is able to grow in the bloodstream where it's constantly exposed to the immune system? How can they grow in these environments and not be cleared really rapidly? And it turns out that they use really what the face of it is really quite a simple approach, but the underlying biology that drives this is really rather complex. So, in essence, what trypanosomes do is that they shroud the surface of the cell in one very dense, what's called a coat of a protein, and the protein's called the variant surface glycoprotein, but the name's not important. It's just a protein that forms a kind of protective layer around the whole of the cell, and it does that in order that basically it becomes the only protein that we as the infected host can see. So all the other proteins that you actually have to get in the top of every cell to make a cell function, to take up nutrients and so forth, they're not seen. Instead, the only thing that we see is this variant surface glycoprotein, this surface coat. And that alone means that it has a short-term capacity to survive, because when we generate antibodies, we only generate antibodies or immune reaction against the VSG and not the other proteins. But of course, the problem is that we do generate an immune reaction against the surface coat, the VSG. And what that means is they have to come up with a means to constantly change this. And in essence, what they're doing by doing this is they're just running away from our immune reaction. So what the parasite, the trypanosome, does is periodically it goes from expressing one surface coat to another, and then another, and then another, and another, and it just keeps moving forward, which means that as the host, when we start to get, it takes a few days for an immune reaction to form against a particular surface coat that's made, and because they're able to change the expression to another surface coat, that provides them with a short window where they can survive. And what this seems to be manifest in infections is that you get when trypanosomes go into the host, they grow up, and you can begin to see the number of parasites grow, and then there'll be an immune reaction against them, and the numbers will drop. But then, because they've changed from one type of VSG, one type of surface coat to another, you get another increase in trypanosomes, and then another drop as the immune reaction comes in, and then you get outgrowth, and it goes on and on and on. And this seems to be able to.
Well, go ahead. The question.
No, it's okay. The only final thing I was going to say is that it seems to be so successful, this strategy, that they're capable of sustaining infections for years and years. And in fact, it looks as though very similar strategies are used in lots and lots of different organisms, bacteria and fungi and so forth.
Well, what's interesting is there has to be good communication between the individual cells of the parasite, because let's say there's 100 of them, and they have, protein code A. How do they know it'll all switch to B? And then once there's a million of them, I would think they all upgrade to the new protein code. Have you observed how fast they do it, and do they do it in concert? And how do they orchestrate this evasion of immunity once they get to large numbers?
Okay, that's a great question, and that's what I've described to you is a kind of simplified version of how this works. You're absolutely right. In principle, what they're doing is they're going from expressing surface coat A to surface coat B to surface coat C, D, and so forth. And you're absolutely right. If that was the case, what you would imagine they're doing is they're communicating with each other in some way to say, okay, now we need to switch from this surface coat to another. Or alternatively, they're monitoring something about the host to say, here's the immune reaction now beginning to kick in, and we then have to make the switch. One of the big surprises over the years that we've been studying this is that there seems to be not that sophisticated level of coordination at all. What they do, as far as we can tell, is that they make these changes in gene and surface coat expression just all the time. Just they do it stochastically and randomly all the time. So they don't actually listen to the immune reaction of the host in order to respond and change coat. They just do it themselves. And equally, they don't necessarily say, I'm now expressing VSG A and I'm going to express VSG B. Once they begin that process, it can move in lots of different directions. One cell will move to B, another to C, another to D, and it becomes an incredibly complex mixture of surface coats that are expressed. So it's a much less tidy process than you might imagine. Now there's one complexity to add to that. It's not the case that the parasites don't communicate with each other at all. They absolutely do. So there's very, very nice work mainly by a colleague in Edinburgh, but a number of other people that show that actually the trypanosomes do know when they reach a high density of infection. And what they do in those cases is they actually turn off replication of the cells. They do that, in fact, to try and prepare themselves to be taken up by tsetse flies. But that almost certainly, and we don't at all understand this, but it almost certainly influences the way this coat switching operates, or it certainly provides another perspective on it. It should be, in summary to what I was saying, it should be a nice simple ordered process, but in fact it's remarkably complicated, the process, and it generates an enormous amount of diversity in the cells, in the populations.
So when you look at someone that's infected, what does the population of various protein coats look like over time? Have you studied that? You have 100% A, or to start out with like 95% A and 5% B, and then once it develops to a certain point, it's 12% of A, 18% of B, 13% of C. Like, what does that look like?
So those kind of experiments. So a lot of the experiments that we do in these to analyze this is using modified trypanosomes. So we'll make mutants, for instance, in particular genes, and then in a culture dish we can grow them, and we can ask, okay, we know at this point when we made these mutants that are expressing VSGA, how frequently do they change to another VSG, and what type of VSG do they change to? What we realized in the last probably 5 or 6 years, and this is from a number of different colleagues around the world, is that kind of experiment in the lab doesn't really capture the full complexity of what's going on. So a number of two very nice sets of experiments have been done recently where people set up or followed the course of infections, and what they did was they used sophisticated sequencing approaches to capture the VSG genes that are being expressed. And what seems to happen there is exactly what you're saying. So what they'll do is you put in a small number of trypanosomes to begin with, and then as you follow the course of infection, quite rapidly you get a really diverse collection of different VSGs being expressed, and that diversity seems to be maintained right throughout the course of an infection. Now, what we don't really understand, and this is where we're leading to in the experiments we're doing now, is is there an order in there? So there's when they express one VSG, when one cell expresses one VSG, does it individually lead to another VSG and so forth? And is the complexity in there because of a population dynamics? So there's maybe order within a single cell, but there's much bigger complexity in a population. And these are difficult experiments to do, as you might imagine.
Well, I would think too, at a certain point, it's got to be communication. I would think at a certain point, the parasite now has to say, all right, we need to make this host excrete something or express something so that the outside world knows we're here and will take us up so we can move on. So there's got to be that happening too. And at what point does that happen? And does that change as the expression profile changes of the parasite?
So that's a very perceptive point you make, and that we now do understand how that works. So they certainly do. The parasites definitely do secrete a signal that causes this change that I was describing in between rapidly replicating cells and non-replicating cells. So a lot of work from a number of different labs has begun to dissect that, and what they do is they seem to secrete this signal. And when you get lots and lots of parasites, you inevitably build up more and more of this signal just by density. And when that happens, it triggers, it reaches a threshold and it triggers a change in the behavior of the trypanosome. So what they do effectively is they take that signal, they take it into the trypanosomes, and that leads to a cascade of gene expression changes that changes their whole biology. It changes their morphology as well, their metabolism, their DNA replication. Now, the question you ask is absolutely correct. What we're now trying to understand is how does that link to the process of VSG code switching, of what's called antigenic variation. And at this stage, we don't have a really clear answer to that. Most of the time when we do experiments, what we're doing is we're doing experiments in actually fairly artificial cells that replicate. They're just machines for replication, and they've lost the ability to secrete or recognize this signal. And part of the reason for that is because we're fairly sure that the process of replication, of genome replication, is intimately tied to antigenic variation, VSG code switching. But the fact that they actually have this added behavior of secreting a signal, responding to the signal, and changing their behavior, it leads us to believe there must be a link to antigenic variation, but we don't exactly know yet what that link is and how it might be manifest, or how the gene expression changes that might occur are linked to it. It's where we're leading to in studies at the moment, but we haven't quite reached that stage yet.
I think, this is just pure opinion on my part, but I think it's better to assume that the parasite, the individual cells, are far smarter than we can imagine, because they need to monitor the host. Think about it. They need to monitor the host to see how sick it is. At some point, they're going to be in trouble. They need to modify the host somehow so that, again, the host will give off a signal that they're there. They can be taken up by someone else. They need to know how many of them there are. They need to know the portfolio of expression of the coats, the immune system invasion, how that's going. It sounds like to me there has to be tremendous communication on many levels going on for this to be a successful endeavor. They have to monitor a lot of things.
So what you're saying is fundamentally correct, of course. It really wouldn't necessarily be to the benefit of the parasite to grow so rapidly that they reach a really high density in the host, and that consumes all the nutrients that we have in our blood, for instance, and overwhelms the host and kills them. Because what that means is that they limit their ability to transmit. And we don't exactly understand to transmit to another host through the tsetse fly, and we don't really know what the time frame of that is. But of course, the longer you can sustain an infection, the bigger the chance you have of being taken up by another tsetse fly that feeds on an infected host. So almost certainly what we were just talking about there, this density signaling mechanism is probably part of that. Is probably a means to cap the level, the intensity of the infection, to try and limit the possibility of killing the host. But it's kind of messy, this process, because we know that other things are going on here. So what I've given you the impression up to now is that trypanosomes live just in the bloodstream, our arteries and veins. Actually, we now know that that's not true. There's been recent work that's begun to explore where you can find trypanosomes in an infected individual, and what we can see is that the parasites move into tissues in the host. So they move into adipose tissue, which is fat tissue. They move into the skin, and in some organisms they move into the reproductive organs. And there's evidence that in those circumstances, their behavior, their morphology, their gene expression, their metabolism changes. And that may be a component of exactly what you've been describing. It might be what they're doing there is trying to limit the potential that they overwhelm the host. So if they move into these tissues, that can provide them with maybe a respite from the immune response. Maybe it provides the host with a respite from the infection, but it maybe also provides a little pool of infected parasite trypanosomes that can feed back into the host. Now, this isn't probably perfect, though, because one of the reasons the disease is called sleeping sickness is because the trypanosomes not only go into skin and fat tissue and so forth. What they also do late on infections is they cross what's called the blood-brain barrier, and they go into the central nervous system. And it's at that point that the main symptoms of the disease are manifest, which are neurological disorders. So it's their behavior. What we're beginning to learn in the last few years is that the idea that they're simple replicated machines in the blood is really not true. They disseminate more widely in the host, and that may relate to exactly what you've been saying, Richard, that they're trying to modify their biology to maintain infections and maintain their host. It might be.
So what? Another question is how much variation has been observed in the glycoproteins, for instance? Is it endless variation? Does it follow a pathway that is followed over and over again, therefore it's predictive? Or what have you seen there?
So there's quite old data that suggests there is a kind of a pattern of the way certain types of VSGs are expressed. So we know that some classes of VSGs are found relatively early infections, and some classes of VSGs are found slightly later infections, and then another class of VSG are found late on in infections. Now, I'm saying classes of VSGs because it's not very specific VSGs, and we don't exactly know everything about what makes these classes. The one thing that we've learned about how this might operate is, and this comes back to what you just said about is there endless variation, is that one of the things they do very late in infections is they begin to generate what look like completely new VSGs that we don't normally see if we go searching in the genome. So let me try and explain this a little bit more. So in any genome, what we tend to think of is genes are there to generate products that are immediately active and do things like they make a structural protein or they catalyze an enzymatic reaction. In any genome, there's a few genes within the genome that we call pseudogenes, and these are genes that at some point or other were functional. They could generate a product that's functional, but they've suffered mutations that means that they lose activity, or they maybe even aren't full-length proteins. They can't generate full-length proteins. It turns out that what trypanosomes have done is they filled their genome with VSG pseudogenes, and what we know is that term pseudogenes in the context of trypanosomes is actually inaccurate, because pseudogenes would suggest they do nothing. They're there just as remnants of active genes. Actually, what trypanosomes do with these pseudogenes is they are a prime source of these completely novel VSG coats that are made. So what they seem to be able to do is they seem to be able to take little parts of all these genes that can be they're often pseudogenes, and they can splice them together in really complex patterns to generate VSGs that really at the start of an infection weren't there in the genome at all, but by the end of the infection they are there. And the scale of this is truly remarkable. So the trypanosome has got, we don't know the exact number, but it's thousands of these genes. If across the whole genome, it's maybe 20% of all the genes that it contains in its genome. And because it looks as though they can do these combinatorial events to splice them together, we begin to think that the capacity for new VSGs is virtually limitless. We're not absolutely sure of that because quantifying these things is really hard, obviously, but it looks potentially like there's a huge, nearly endless capacity to make new VSGs. And there's a parallel to be drawn there between what the trypanosomes do and how we as the host fights the infection. So I'm sure yourself and lots of your listeners know that what we do is our immune system is generated in the course of development, and what we do in the course of that development is we take genes that make the important antibodies, for instance, or the receptors for the immune cells, and we rearrange them and we build them up into thousands and thousands of novel genes. And that is the mechanisms that underlie that and VSG switching are very, very different, but the principles may well be the same. That what you need to do what we need to do to tackle infections is have a really sophisticated armory of immune reactive molecules. And what the trypanosome has to do to counter that is have a really wide armory of these surface coats. Trying to quantify this and trying to quantify VSG switching and trying to quantify the numbers of these is what we're aiming to do now, but it's a big challenge.
How many different types of animals and creatures, for instance, can the tsetse fly infect? And have you compared the expression of the different protein coats in various animals to see if, like, in certain ones, there's a certain pattern, in other ones there's another pattern, or even amongst humans?
Yep. That's okay. So that's actually, this is what you're asking there is really a key question in how the process of antigenic variation operates. Because what I've really been talking about here is, in fact, one species of trypanosome. It's called Trypanosoma brucei. And most of our knowledge of how this works is derived through study of Trypanosoma brucei. And in part that's because it was one of the first trypanosome species that was found, and it was the first that was able to be cultured in the lab. It was the first that we could genetically manipulate. So it became the kind of workhorse for how we do things. Actually, trypanosomes infect lots of different species. And Trypanosoma brucei, some species, are largely infect humans. There's a number of other species of trypanosomes that really don't infect humans at all. They infect animals. So examples of that are called Trypanosoma vivax and Trypanosoma congolense. And we, until a few years ago, I think we were convinced that all the lessons that we've learned in Trypanosoma brucei would be easily converted to the two other species, which are very closely, in evolutionary sense, are very close. But experiments that are now being done, so just sequencing the genome of these parasites, trying to catalogue all the VSGs that are in them, and experiments that we're beginning to initiate now are beginning to suggest to us that it might not be the same at all. They may use antigenic variation, despite the fact they all have VSGs. The catalogue of VSGs that they have is different, and there's some evidence that the way that they rearrange those VSGs is very different. So we may have fallen into a slight trap in thinking about exactly how things work in Trypanosoma brucei and imagining this is absolutely the case for other trypanosomes that are similar to it. So we need to be careful. We need to test a lot of the things that we did in Trypanosoma brucei. We now have to revisit in these other species to try and really understand the underlying game that's going on effectively.
Why can't you inoculate someone? Why can't you get a population of these parasites that have expressed 500 different glycoproteins, kill them, but still have the proteins intact and inject them into someone, whether they're infected or not infected. Maybe someone that's recently infected, you can do later stage population that's been killed and inject that into them to inform the body to stop it, or vice versa, or someone that's not infected at all.
So one of the reasons people regularly say that you can't generate vaccines against trypanosomes is because of the system of antigenic variation, and it's because the thinking there is that what you would want to do for a vaccine is immunize them with a protein that's very conserved, so that when they're confronted by any different trypanosome, they're capable of recognizing that, because that protein, that antigen, is really similar between the different trypanosomes. The thinking behind that is the problem is the VSG coat prevents recognition of those much more conserved antigens. So the question you were asking is an interesting one. So could you actually simply understand the entire repertoire of VSGs that are ever possible of being made and generate a cocktail that captures all of that? So in Trypanosoma brucei, which we've been talking about, I think the answer to that is probably no. And the reason is what I've just described. They're capable, it seems, of virtually limitless rearrangement of these genes so that they can generate very, very novel VSGs that, however hard you try to generate a collection of 500 or 1,000 different VSGs as a source of immunization, they're still able to generate new ones. For the other trypanosome species, I think at this stage we don't quite know that. If they use a much more limited repertoire of genes, or they're not capable of these really complex rearrangements, maybe then there is the potential to do that. But no one's been brave enough to try that kind of experiment yet at all.
I'm trying to think of what I was going to ask you here.
No problem.
Not everyone dies of this infection, right? I mean, there are people that build up. What happens? What have we learned about studying people that survive this? What is their immune system doing to eventually get rid of the parasite, or does it just go into like a dormant state forever and still stay resident with them? Like, what are we learning from there?
Well, in fact, in virtually all cases of a human being infected by a trypanosome, if you don't intervene at all, it will be lethal. So most of the studies suggest that once a person is infected, if you don't provide drugs to try and clear that infection, they will ultimately die. There are small number of cases where there has been reports of people who have been infected, and then through long-term monitoring, they don't seem to have trypanosomes. Now, whether that means that they really have successfully managed to completely fight off the infection and they're absolutely clear and they never get infected again, or whether the trypanosomes are present in those individuals and we've not been able to detect them and the disease might reoccur much later on, I think we don't know. And the reason for that is because the infection can last for such a prolonged period of time. But the basis of people dying or animals dying of trypanosomiasis is not really to do with really long-term infections. It's actually to do with the movement of the trypanosome from the blood into the central nervous system. And when it gets into the central nervous system, its effects, neurological effects, are so severe that that's the cause of the disease. It's actually not. If it only lived in the blood, probably people could they would be ill, but they could live for a really prolonged period of time with it. So we can't really there are so few cases of people that we know have been infected and are apparently clear that it's very difficult to try and understand what might or might not happen in those individuals.
What's just a couple more questions on this. I know
no problem.
There's tons to look at.
Yeah.
Where do you think the breakthrough's going to come? I know, if you knew, you'd be further along. But what are your guesses? What are 2 or 3 promising leads that you're following up on where you think a breakthrough is going to come, and what will that breakthrough be?
So I think one of the really in this area of the way the trypanosome infects and the dynamics of the infection and the transmission, what we were talking about as regards the communication between them, where they change from replicative to non-replicative forms. I suspect that has real promise in thinking about trying to intervene in the disease, because that's likely to be a quite specific process, a process that's evolved specifically in the parasite. That means if you can really understand it and you can interrupt it, you can probably block the prolongation of the infection. You can probably cure somebody with that. What we're becoming interested in now is so we sort of understand how the rearrangement reactions that occur between VSGs occur. So we sort of understand the machinery, the cellular machinery that drives those reactions or catalyzes those reactions. And the sad thing about that machinery is it looks like it's very conserved with us. It looks as though it's just a general system for maintenance of genomes. So what we are now beginning to try and understand is there something at the very head of the reaction, the very top of the reaction, that's specific? So how do they actually start the process of changing from one VSG to another? And when I say we, it's not just my lab. It's a number of other labs that are doing this. And in this case, there are some potentially promising leads. So recent studies have found what looks like a very novel factor that seems to interact with the site that the VSG gene is being expressed in, and it modulates its activity. And that looks really, we don't know much about it, but it looks very specific to trypanosomes or trypanosome-related organisms. So that could be a good lead. And what we've been trying to do is trying to characterize the actual genome damage events that might start this whole process of rearrangement. And in that case, we're beginning to get some leads about structures that form where the VSG is being expressed. That again, if we could really understand what those structures are, what they're bound by, and how they're turned into the rearrangements, again, that might be a lead for intervention. But it has to be said at this stage, these are kind of basic fundamental science questions. They're not quite at the stage of translation into drugs, although with luck that could be possible.
Well, very good. So what's the best way for people to find out more and to get in touch with the lab or you?
So people are very welcome to ask questions on Twitter. So the lab has got a Twitter page that we're more than happy to interact with. We've also got a lab website, and my lab is part of a bigger consortium of parasitologists in University of Glasgow, some of whom are in what's called a Welcome Centre for Integrative Parasitology, and that's got a website where they'd be absolutely more than welcome to email through the administrative staff, and they'll disseminate any questions they've got about parasitology to myself or any of my colleagues.
Actually, you should do an unwelcome center for parasitology because parasites are never welcome.
That's very true. Very true.
Well, very good. Well, Rich, thanks for coming on the podcast. I appreciate it.
Okay, thank you very much for listening to all I had to say. It was very nice to talk to you.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you.